I didn't volunteer to come here. I was requested, so I don't run away from responsibilities. That wanted me here, so apologies for carrying out these sessions. Anyway, let's go for the final session, which looks like going to be very interesting. Senior professors. Let's start with Professor Iqbal talking about plant and environmental science. Over to you, Professor.
Good afternoon, everybody. Okay, so good afternoon to all of you. This is the last session, and I'm the first of 8 speakers, so you have some time to tolerate till you go home. So my talk is on plant and environmental sciences. And we have 3 ongoing major projects, that is plant culture, environmental remediation, and the role of climate society and climate change and diseases. So the first one is on plant tissue culture, and this is my research assistant Mandala, who is doing all the hard work. And basically, Sri Lanka has 30%, nearly 30% cover of forest, total cover. And of this, a little more than 50%, 53% is dry forests. The problem with dry forests is that dry forests, the seed production in dry forests is seasonal, and the seeds are also slow growing, they have dormancy, and therefore reforestation is a problem. And why reforestation? Because the dry forests, unlike the wet forests like Singharaja, the dry forests are being cut down for conversion to agriculture, for harvesting the timber, human interventions, etc. So to overcome this problem, we have been looking at how we can contribute towards this reforestation. And we looked at 2 things. One is to enhance the natural propagation of the seeds, and also micropropagation to produce new plant material. So those are right on top, you can see the 3 species we are working with. And we did very simple thing to overcome the dormancy. And that is culturing the seeds in vitro under sterile conditions, but by removing the seed coat, because the seed coat is very thick, hard, and that prevents germination usually. And remove the seed coat, did in vitro culture of the sterilized seeds, sterilized embryo, and through this they were able to generate seedlings. We were also able to generate seedlings by applying gibberellic acid to also overcome dormancy, in contrast to mechanical scarification, etc., to overcome dormancy. The traditional tissue culture methods for micropropagation, but here again, the major problem we have is inducing the plantlets which we produced to root. Rooting is a major problem in tree species. We also looked at propagating America, which this is in copricide, which is very sweet, in fact, over 200 times sweeter than sugar, no calories, and therefore it's an ideal product for the pharmaceutical and food industry. But the problem with this plant is, again, the seeds are incompatible, there's incompatibility of the seeds, and therefore you can't propagate it through seeds, you have to use cuttings, and therefore we looked at how this can be done through plant tissue culture, and we were able to develop a protocol which produces plantlets within a space of 8 weeks. So with this, from the time we make the cuttings, sterilize them, and also of course tissue culture is an expensive process, and therefore to overcome that we used a very natural supplement to do the tissue culture, that is by using coconut water, which coconut water is of course sterile. So using this, we were able to develop a protocol to produce plantlets in 8 weeks. That's about the plant tissue culture project. Next is the environmental remediation project. And here the major work is done by my research assistant, Prasika Visanayaga. He completed his PhD recently. And this other pretty lady you see is the undergraduate. She has also finished a BSc degree. The previous years, we were looking at how we can remove pollutants from the waterways, from aquatic systems. For example, heavy metals and also textile dyes. Last year, we moved on to a new field initiated by Rasiga, and that is to fix carbon dioxide. Now, he had an innovative idea to fix carbon dioxide using what are called metal organic frameworks. Of course, nature does it very much more elegantly in the leaves that you see outside. Through a series of enzymes and electron donors, the CO2 in the atmosphere is fixed into various carbohydrates. So to mimic this, of course, with this issues of climate change, is necessary to pull out the CO2. We looked at how actually Rasiga came up with this proposal to use this metal organic frameworks, which actually have crystalline structures, highly porous, and therefore high surface area, how they can be used to fix CO2. So initially, the experiments showed that we could fix CO2 and produce a very basic organic acid, the formic acid. And with this simple experimental setup here, he was able to show first qualitatively from the diagrams here. These showed that there was an OH and a CWH group produced. This was quantified later using HPLC. And this very tiny peak shows that its formic acid was produced. This, of course, was a small scale batch experiment, and this is going to be upscaled. And this is a collaboration with the National Science Foundation and also the German Academic Exchange Service. So there are scientists in Germany who came here to do the work, and our scientists also go to Germany. But because of this pandemic, they have not been able to go, probably sometime in the future. So this work has potential, which we carry forward. The next one is phosphates. Phosphates is another major environmental problem for the simple reason that the phosphate cycle is open-ended. Phosphates we can mine from phosphate rocks. It is applied as a fertilizer, and after that it is ultimately washed into the sea where it is sedimented. Unlike the nitrogen cycle or the carbon cycle, which is so here, unless we recover the phosphates we are using, in about 100 to 200 years, all our phosphates will be mined, and then we know phosphates. And of course, phosphates is very important. It's the backbone of the DNA molecule, and it's also an important non-nutrient. So therefore, we looked at how a simple way of recovering the phosphates, and what we did was we screened aquatic plants which can take up phosphorus at a higher concentration and converted this in a dry form into a fertilizer. We used this on moonbeams and found that it can, to a very large extent, replace the conventional triple superphosphate as a fertilizer. So we can, in the future, we want to see to what extent this can replace the conventional fertilizer to reduce the fertilizer input from inorganic sources. So I'll just take a few seconds to complete this. This was part of the NRC mega project on dengue. We were part of that. The 5 years is just being completed, and my student also, he has completed his PhD last year. And the last part of his project was how we can develop larval indices to predict an onset of a dengue epidemic. So by coming out with household and what's called a Brito index, he was able to calculate indices, and these showed that we can predict the onset of dengue at least about 2 months before the onset of the epidemic, particularly in Colombo and Kandy districts, at 3 risk levels of low, moderate, and high risk. I'll stop there for any questions that you may have, and that is of course not part of that's the science dissemination that we had, and of course that's more or less my last slide. And you can ask questions. Thank you.
We have time for one question. Thank you very much, Professor.
Yes.
What is the MOF material you use to get formic acid?
This has the metal ion is the metal ion is ferrous, and I'm sorry. Yeah. So the metal ion is ferrous, and we used organic ligands, and also we used a very green synthetic method by using
Are you sure that formic acid was produced by reduction of carbon dioxide?
That is what is shown here from the HPLC.
It's more likely that formic acid was produced from degradation of MOF. Photodegradation of MOF. Most MOF are not very stable towards UV, and they degrade, and during degradation, formic acid is a very tightly compound. So you have to test.
We'll have to run another control for that.
Yeah.
But in which case also, sir, it is probably, if that was the case, there would have been a very high level of formic acid also been produced.
You can't say. I think you have to do a confirmatory test.
Yeah, I'm not
I
sure.
find out whether it, because we did so many experiments long time ago trying to reduce carbon dioxide. It was very difficult. Every time moment you use an organic compound, things like formic acid and aldehydes and all these things are formed because UV degradation is more likely than reduction of carbon dioxide.
Yeah, so we'll look at the organic linkers.
I am not saying it is wrong, but you have to do it.
Yeah, we'll certainly test that. Yeah, we'll run a control without this and see how it is. But of course, I should also add that many groups have reported the formation of not only formic acid, in fact, even higher levels of even methanol. Okay, they have gone up to methanol.
Yeah, all kinds of reports. Producibility and we confirmate test is very, very important in things like this.
Yeah, so we'll run a control and check on that also.
Thank you, Professor Tenapal. I think this discussion should go ahead even after this meeting. I mean, this is the sole purpose of presentation. I had a question, but I'm not going to ask. But could you kindly share that paper with me? Because to me, the most important thing is this dengue study prediction, which has a huge potential for impact.
Yeah.
So I'll stop there.
Okay, thank you very much.
Thank you very much.
So now I would like to invite Professor Lakshman Disanayak to give a presentation on condensed matter physics and the solid state physics here.
Good afternoon, everybody. I have only 40 slides, I think can manage. So, this is a progress during 2019 condensed matter physics and solid state chemistry project. We are focusing on alternative energy sources, out of those, solar energy. Oil, as everybody knows, will last only for about 40 years, gas 65, coal 155, but all these three will pollute the environment heavily. So people are turning to more and more alternative energy sources, mainly solar and wind. But up to now, it's very highly underutilized solar electricity globally. And in Sri Lanka is worse, 0 point something. The US 2%, Germany, smart enough, 10%. That's the world highest user of solar energy. Solar energy, you can have a thermal route to heat hot water, or you can have a photovoltaic route to generate electricity, again in two ways. Either you fix the solar panels on rooftops, or you can have a large number of such panels in a large solar farm. Now at present, all those which I showed you are made of silicon material, silicon PN junction semiconductors. Can be crystalline or amorphous, two versions. So commercially available, you can buy Only problem is it's still expensive compared to grid electricity. And there are new solar cells are coming up, emerging, almost commercialized. Cadmium telluride, one version of thin film solar cells, copper indium diselenite, high efficiency, perovskite, the latest. And in our labs, we are doing most of our work on dye-sensitized solar cells, which can be categorized as the third generation. Many more years to go, at least 5-6 years before it's mature and become commercial. The action of a solar dye-sensitized solar cell, you have an electrolyte, typically say iodine, iodide, triiodide electrolyte, and titanium dioxide photoanode, which has to be sensitized with a dye because it's a wide bandgap semiconductor. So to catch photons, you have to dye sensitize the photoanode, and then on the other side you have a typically platinized copper electrode. And during 2019, we have studied 3 systems. One is a substitute for the dyes. These dyes are generally expensive, pneumatic dyes. So alternative method is to sensitize with quantum dots, semiconductor quantum dots. That's one of our projects during that year. And also you can nanostructurally modify the TiO2 photoanode using higher light scattering techniques. That's the project 2. Project 3 is try to improve the electrolyte. Now in this particular system, polyaniline has been incorporated into a polymer electrolyte. All these 3 will increase the enhancement of dye-sensitized solar cells. This is a system, is a tri-layer. This has 3 layers of TiO2, and the central layer is sensitized with colloidal nanoparticles, quantum dots, to improve the efficiency. This is a second system where TiO2 photoanode is modified by incorporating a scattering layer to catch more sunlight. And this is the polyaniline incorporated electrolyte, PSSC. All these 3, you get at least 1 to 1.5 or 1 to 2% increase in efficiency. These are the postgraduate students being trained. I have 2 RAs funded by the NIFS. Third one is to be filled. Then we have externally funded 3 RAs. One from University of Jaffna, one from Southeastern, one from Open University through a HEAD project. These are the key publications, 1, 2, 3, 4. 3 are in ACI journals. These covering 2019 work. So many abstracts. I don't want to go into details. With the Jaffna conference, international conference in Jaffna, PGIS, University of Peradeniya. This is also Young Scientist Forum in IFS. International Conference. These are extended abstracts. Indonesia Conference, international abstracts. Malaysia International Conference, extended abstracts. As invited speakers, myself, and visiting professor here, Professor Senadira. Research collaborations, there are 5 active collaborations during 2019. One with Sweden, the long-term collaboration, one with Department of Physics, Peradeniya, then one with OLS, and also with Jason here. Postgraduate students wise, one MPhil was graduated in 2019. Undergraduates, 3 undergraduates completed their projects in that year. 2 from SJP, 1 from Eastern University. Research grants, 1, 2, 3. These are new grants, 2 by Dr. Senadira, 1 by Mr. Senthuran from University of Jaffna, small grants. These are continuing grants, 1 with Sweden, 1 from the ministry. It's a multi-university national project in developing solar cells and other aspects. Then NSF 2 grants. National development, member of several committees continued during that year, reviewing journal articles, invited speaker. Those are the national contributions, and we participated in national exhibitions. This is a BMCH exhibition where I was leading the renewable energy theme, tech theme. Of course, with the assistance of many other people, Percival and others. NRC Merit Award 2019. Membership of boards and committees. Okay, this is this is part of the exhibition. Photos, BMCH, demonstrating the solar energy devices. These some of the school science program activities here. Okay, this is part of the awareness program of solar energy conducted nationally through a multinational, multi-university project funded by the ministry. Awareness programs for O-level and A-level students to highlight the importance of renewable energy. We had awareness programs, hands-on sessions, essay competitions, and these are the schools covered by the night. These are some of those photos. These are conducted mostly at Peradeniya University from nearby schools, Novara Central, Rampitia, Ranabima, Lampara schools. And the students enjoyed very much the fascination of using solar energy for lighting, for turning motors, for water pumping, many applications. So they are the messengers. They take this message to their parents. So this is an indirect way of educating the community on the importance of renewable energy. The same thing, school programs and technician training program. In Sri Lanka, up to now, there is no accredited program to train solar technicians. So we drew up the syllabuses, and the program has started already with NITA and Sustainable Energy Authority to train at least 500 solar technicians. About 200 already trained, because they will be highly in demand in the years to come. Yeah. Newspaper articles, Sunday Times, don't let them sit off solar power, because government, especially the CEP, is very reluctant to popularize this solar energy due to some reason. So we have to write to newspapers to convince the authorities. But now the new government has adopted 60% from fossil fuels, 40% from renewables by 2030. There's another project, nanofilters for water purification. It's an ongoing project, very slow. We don't have a research assistant. These are research group in the lab, outside in the garden. The true sign of intelligence is not knowledge, but imagination. Everything which is new has to come from fundamental research. Otherwise, it's not new. No country in the post-colonial era has thrived without first building its capacity to conduct scientific research. Thank you.
Thank you very much, Professor Lakshman. It's an excellent presentation. Actually, we don't have time for the question, but I want to compliment the connections, like not only the research and your help to the institution development, and also your engagement with the public is a really remarkable. I think really thank you very much, and I really acknowledge and appreciate that contribution. Thank you.
Thank you. You can organize the special award next year.
Professor, I have a burning question. That's a burning question. When I was looking at them, this is something bothered me throughout. The panel is like this.
Yeah.
So when sun sets, it goes halfway through.
Yeah.
It doesn't capture. So my question is why it's not like this, why it's always like this. That have been bothering me for many years.
Because I think when you slant it towards the east, you catch a lot of sunlight or where most of sunlight. On the other half, some people usually fix panels facing east, facing west. Capture.
So let's invite now Professor Kulasurya. I always say this, I'm listening to him presenting today, but his thesis when I saw I was 17 years.
Good afternoon, Chairman, ladies and gentlemen. I'm just going to do a very brief resume of our rhizobium project. The background is that the mission of the project is to improve nitrogen fixation in crop legumes by rhizobia biofertilizer inoculation and minimize the use of chemical fertilizer in legume crop production. So the basic research part of this, to achieve this mission, is to isolate, characterize, authenticate, screen and select crop-specific best strains of rhizobia, which are soil inhabiting microorganisms, for biofertilizer inoculant preparation. So that takes a lot of time, and we have, of course, worked for years to come to this stage. And then once you select the strains, you have to develop and field test them in small scale. Field plots to see whether they do the same thing as we find in the laboratory in the field. And then that's another stage of screening. Select the best out of those, then convert them into the inoculants that give the best crop growth and crop yields. And always we can compare them with the nitrogen fertilizer used to see how much of fertilizer can be minimized by way of inoculation. Then, of course, you have to transfer the technology to the end users, the farmers, that needs a lot of effort and energy because they are inculcated from the 1960s, no fertilizer, no crop production. So we had to change their mentality. This is what we, just going back, just to give you an idea what we have achieved. And these are pictures of different crops we have inoculated in the final stage after all the selection. Application in the field, these are the results. Soybean, in fact, these pictures have come from the Department of Agriculture. They used our inoculants to do extensive cultivation of soybean, and they got these wonderful results. They gave us a list of 100 pictures we have selected from that. Then the next one is vegetable bean, and then you have mung bean, one of our field officers checking with the farmer the nodulation of the field bone, and the forage legume, clover, grown on the farm. Then we went into some of the basic studies. This from here onwards, I am concentrating on what we have done in 2019, what you saw in the previous slides, so background work. Now, we have been using these inoculants for field application, but we have not had the opportunity to characterize them at the molecular level. This opportunity we got because we got a student co-chaired and co-supervised by Professor Rajapaksa of the University of Pennsylvania, Biotechnology Department, and she performed, because we don't get any grants, we earn our own money, and we didn't have enough money to cover the entire lot of rhizobia gene bank we have. So we selected those rhizobia we have field tested. That was only 19 rhizobia, and they were a DNA analysis and fingerprinting was done, and results showed that at 70% similarity coefficient, you get it, they separate to 10 clusters, and then furthermore, then we use only 5, because we use 5 of the strains that are used. Now we have released for farmer use for the 5 crops. And those 5, the 16S RNA analysis was done, and after most likely 3 analysis, they were identified into the genus level. And this program is continuing. We have now got another student to go further into more detailed analysis of molecular analysis to really identify that to the species level. At the moment, we can only talk about the genus we are using as inoculants for the different crops. That's the molecular characterization. So what we got there was the one that we are using for soybean, which is just given a number as S13, is a rhizobium species. For green gram, baby rhizobium species. Groundnut is a cynorhizobium. As far as we know, this is the first record of cynorhizobium in Sri Lanka. Vegetable bean, a rhizobium species. Clover, also a rhizobium species. But this is a preliminary analysis. Then the next thing we did in 2019 was to set up a field experiment in Ankumura, and as a part of the training, together with our staff, this was a student, Kokila Anuruddhi, from Ruhuna University. She came to do a student project, not postgraduate, but graduate student, undergraduate student project. And she participated in this field experiment together with our scientists to see how much of nitrogen is replaced by our inoculant for vegetable beans. Because vegetable bean inoculants that we produce have now become extremely popular in and around Candy. In fact, we are collaborating with the Candy Provincial Council Agriculture Department, which has now started distributing, purchasing from us and distributing our inoculants to the farmers from Hamuranketa to Valimata, Bandarwala area. And this experiment, we conducted the recommended level of fertilizer, urea, nitrogen fertilizer, recommended by the department for a bean is 220 kilograms of urea per hectare. It's a lot of urea. And so that was our maximum. Minimum was 0. And we get half that amount, quarter of the amount, 3/4 of the amount, and we looked at the yield produced by the application of urea. So it's what you call a curve fit. This is how the blue line shows how the yield has responded to the nitrogen fertilizer. That means this crop is, of course, responding to nitrogen. With every little bit of nitrogen given, it increases yield up to that top level recommended by the department. Then we use our inoculant. The black line shows, in terms of reading the performance of the inoculant, this goes, and that is almost equivalent to the complete statistical. There's no difference between the entire recommended value and the yield obtained with the inoculum is equivalent to what is given by the full amount of recommended unit of fertilizer. Then, of course, all our extension work, a lot of work has to be transferred to the farmers. This shows our field officers going and discussing and presenting with farmers, workshops, and so on. And that slide shows our extension, the amounts given, and the earnings that we have got is equivalent to, or the savings of urea fertilizer is equivalent to 18.6 million rupees. This is our team who worked with me to achieve this. This was 2019.
Thank you very much, Professor Plesuri. So now question time. One questions. Can I ask one? Okay, Professor Plesuri, when you have select number of strains, that very specific to mung bean, soya bean, and so on, like a very. Did you use a molecular recognition, or how did you select? This will goes into the soya bean.
No, it's like this. We isolate from the targeted crop to begin with, which is growing naturally from different locations. That's why we end up with several strains. In fact, in our culture collection, we have now 150 strains. So each crop growing naturally in different locations, we go and bring that to the lab. And so when we start for mung, we start with the mung nodule and go on isolating and purifying, and then doing testing for best nitrogen fixation, rapid growth, and nodulation in the greenhouse first. Then the small fleet goes. And this screening process narrows down the strains. Finally, we end up with 1 or 2 strains that we are prepared to give to the farmers.
Thank you. Thank you very much, Professor Plesuri. Excellent presentation.
May I invite Professor Bandara to talk about energy and advanced material chemistry. May I also remind all the speakers, if possible, this time confined to 10 minutes. Otherwise, it's 4:15 already. If we take 15 minutes, there are 5 speakers. We would have another 1.5 hours. It would be late, and it would be unreasonable for the last speaker. So, Professor Bandara, the bell will ring at 8 minutes. Then you have 2 minutes.
Okay, we'll try. Okay. Thank you, Chairman. Good afternoon to all of you. The broad theme of the energy and advanced material chemistry group is to harvesting of solar energy. Under this broad theme, the project conduct several research projects. The first project is water splitting, the hydrogen production via water splitting, because we know that fossil fuel is depleting rapidly. So if we produce hydrogen by cheap method like photochemical methods, it could be a future energy source. Also, we do reaction on the carbon dioxide reduction to convert carbon dioxide into methane, ethane. But as Professor Sendagam just now mentioned, it is a very difficult task. I remember even as a graduate student under him, we carried this reaction, but still we are continuing because of this importance. The world has not stopped doing research on carbon dioxide reduction because it's important. Even if we can achieve this reduction, it could be also a mitigation for the greenhouse effect. And the other project that we do research is quantum dot solar cells and thin film solar cells to convert solar energy into chemical energy. And also we have NSF and NRC funded 2 projects, and like fabrication of membrane to separate oil water in surface station water. So in the first project, the hydrogen energy project, we have to break the water molecule into oxygen and hydrogen. We can use 2 different methods. We can use either photocatalytic methods or photoelectrocatalytic method. In the photoelectrocatalytic method, we have several options. Either we can use photos assisted method or direct photoelectrochemical cell. The issue is here, the major concern is to have higher solar to hydrogen conversion efficiency. all these factors are depending on the anode material and the cathode properties of this electrochemical cell. So we are mainly concerned on the properties of photoanodes and the photocathodes. In the photoelectrochemical methods, we developed a copper zinc tin sulfide as a earth abundant material because this crystal structure can absorb light up to 800 nanometers in the visible solid spectrum. The problem is, according to this phase diagram shown in the here, this crystal structure is formed under a small window with a slight change. The other secondary phases are formed, and normally these secondary phases are not good for the good solar cells. But fortunately, we have adapted. Now we have developed system, thermal evaporation and e-beam system. That we have given new system. With this system, we were able to control the crystalline properties of this crystal structure. Using this, we can produce very good films, and you can see that these films, they absorb the light from 800 to the whole solar spectrum. With this system, we tried the water split. Okay, so we have characterized this film by the high resolution TEM method, and we identify all the crystalline phase formed during this different parameters. We have changed the concentration and the preparation methods. By this system, we have optimized the properties, and we have identified all the crystalline phases. And you can see that with this system, we can put the hydrogen production is studied by IV curve. So we have optimized this one, but we have further study this, further improve this system. But the first paper is produced, already submitted in this one, but we'll continue this work. So the next project is the photocatalytic hydrogen production. Normally we use the photocatalysts like semiconductors, but most of the stable semiconductors are wide gap semiconductors. They cannot harvest solar energy efficiently because the energy gap is high, so they absorb only the UV light. So to absorb visible light and the near IR light, we have to narrow down the band gap. So we adopted this to create a interband gap state in this material. For this, we have used strontium titanate. Actually, it has been shown that when we remove the oxygen atoms, so density of states calculation by the mathematical, it has shown that the interband states are generated according to this density of state. So what we use a reduction of strontium titanate by the sodium borohydride under vacuum under pressure, and we can remove this oxygen atom, and some band gaps are created, and we can see that once we create from here to here, we increase the temperature, you can see the absorption of the light. So okay, so we have characterized these cells, the properties by SCM, and also we studied the hydrogen production rate. We can see that, and this is on the pure water. The 550 one was the best because the highly reduced one, and the 350 is not good. And also we have studied the transient kinetic studies, transient currents study to explain all this behavior, but I will not go through this. Okay, so the other one is the quantum dot solar cells. So we have quantum dot solar cells. Advantage is that we can manipulate the solar absorption spectrum by using different quantum dots. They can assemble in a layer wise. We have already, last year we achieved 6.5% efficiency with the three-layer system. So we are continuing with the new system like using antimony sulfide. We have optimized the system one stepwise, and we have achieved so far the solid state one with the 4 point something. So we are still continuing this project. And also this is the NSF funded project to treat the wastewater in the substations. So we have already developed the middle scale reactor, and now we have already developed a large scale reactor. So we install them in a real substation in Kandy and Peradeniya, and we'll check the water quality. Okay, so the output is that we have published 2 papers and conferences, and we have several students trained. Few students, and also we have 2 grants. And last year we got several NRC merit awards, and this is my citations for the total citations. And also I am an editorial board member in the Progress in Photocatalytic and the Front in Chemistry. Oh, this is my research group. Thank you.
We have 30 seconds for one question, quick.
Getting an efficiency about 6 for the quantum dot solid state is quite good. It's very impressive, but how is the stability?
We are studying the stability because if we seal it properly, the sealing is the major problem in our case, because we don't have proper sealing method. But if you, for a certain extent, we will success, but like as a dissensile solar cell, the sealing is not proper. So it goes down, slowly goes down, but that part we have to do actually.
Nonetheless, 6.6 is very good. Very impressive.
But even the other one is actually much better. It's still now it's 4 point. It's more stable.
More stable.
Yes. Still not optimized actually. Those the only 2 parts is optimized, and we have to like back and forth. Optimize it.
Thank you very much, Professor Bandar. It's an excellent presentation. Now I would like to invite Professor Dittis to talk about primate biology project.
Ladies and gentlemen, before I talk about the specifics of this research, I'd like to put this in a general context. We're scientists. Why are we scientists? Certainly not to make money. Some of us are driven by the nature of the phenomenon. We're inquisitive. We're turned on by the new information that we find. We want to share it with our fellow man. And some of us would also like to maybe improve the world just a little bit from the knowledge that we've gained. If you're part of the last group, then you may be disappointed because this is a slide. It takes eons of time before your new discoveries make any impact on the world itself. Let's just take the top example here in this slide. This is, scientists have discovered the problems of mercury as a poison for work 100 years ago. It takes, what, 60-odd years before it's even recognized or acknowledged by the administration, by the governments. Some legislation was approved. It took another 60 years so before there was actually improvement. That's just for mercury. All of the other things these are other sort of threats to our health or the environment. Let's look at climate change as another example. For more than 100 years, people have been talking about the effects of the potential negative effects of climate change on the world. Okay, hitting the wrong button. And for climate change, lots of talk for 50 or 60 years. And a little bit of legislation, but as yet, no green line, no positive effects at all. Climate change, and when we talk about biodiversity, I think we fall into the same sort of trajectory when we talk about biodiversity. Now, what is biodiversity anyway? What are we talking about? My objective here is to focus a little bit on the biodiversity that we have amongst the mammals in Sri Lanka. and to identify some of the natural habitats that are suitable for conservation, and hopefully the canvas for the implementation of some meaningful conservation legislation, which is beyond my ken. But what do we mean with biodiversity? Biodiversity really means the number of species that we have, the numbers of individuals per species, and their geographic distribution. That, in a nutshell, is biodiversity, or at least that's what the popular notion is of biodiversity. There is, of course, another element, another dimension to biodiversity, and that is the variation within a species. And if you talk about the conservation of biodiversity, you really need to look at the diversity within the species, among the subspecies, among populations within a species, because that is the crucible of evolution. That is where new species are evolved, or they are actually also preserved. And as an example of this is a purple-faced langur. Most of us know that it's a species. And it's endemic to Sri Lanka. It's a great monkey. It's interesting. But what few of us know that there's a highly diverse subspecies. There are 4 subspecies, dependent upon their morphologically distinct, probably genetically distinct, genetics hasn't been studied really, and they're geographically distinct. Another favorite monkey of mine, well, so some of them are recognized as subspecies, but within these subspecies, there are even more variants. Here's a white monkey from the southwestern purple-faced langur. I mean, should we preserve this kind of biodiversity? And in order to preserve it, we need to preserve the habitat. I will not bore you to death. These are my favorite monkeys, of course, Macaca sinica. There are 3 subspecies, and they're very distinct by their head here, by their morphology. So the idea here is that within species, there's quite a bit of variation. This is just an outline of the many different species that inhabit the southwestern lowland rainforest, and the number of that are resident, the number that are endemics. This is sort of a long list, which I won't bore you with, but let's look at some specifics. Just focus it. This is just looking at the biodiversity amongst the shrews, amongst the primates, and the degree to which they are actually threatened. I mean, half of these animals are threatened. These are threatened with extinction. Some of them critically. The ones that are in red here, these are critically endangered. But this critically endangered list was not developed by me. This was done by the Ministry of the Environment with David Kavirokun leading the charge. This was a book that was published by the government. Okay, so just looking at the small list of shrews, the small list of mammals, half of them are threatened, for God's sake. Bats. This was totally a surprise to me. Why would bats be threatened? I mean, these animals live up in the treetops. There's no interest in bats, but 50% of these things are actually threatened with extinction. And why? Because of habitat loss. You may not even worry about bats. Now with COVID-19 in our environment, we may think a little bit more about positively or negatively about bats, but at least you have to think about bats. Putting it all together, about 59% of Sri Lankan mammals that inhabit the lowland rainforest are threatened with extinction. And that is dismal, really dismal. And it's just a warning sign. I'm just a messenger. And what do we do to protect us against this? Well, what has been undertaken, of course, we have nature reserves, or we have national parks, and such as the listed here, the Singaraja, Talavate, and so forth. But you know what? It's only less than 0.5% of what lowland rainforest there used to be. 0.5% remaining. And even that 0.5% is threatened. What's going on here? I mean, this is drastic. You can't have this. Wake up, people. And I mean, you have the Singaraja, that has boundaries, but even that is being approached upon. And Vijayasundar and others, we would tell you more about that than I would. So what do we do against it? What can I do about this as a scientist? Well, there's not much I can do. I'm just a messenger. But one of the things that we need to do is enlighten the public. And one way to do that is, okay, these scientific publications that we have, they're in a library somewhere. I share them with you here. Okay, this is as far as it goes. It doesn't reach the people. The only way that you can raise public awareness is through films. Now, the last 2 years, we've made 10 different documentaries. And these are not things me going, these are not films where I run around with my video camera. We've been able to attract the best producers in the world, the BBC, the Terramata from Europe, the Japanese television, the South Koreans. These films have reached the living rooms of millions and millions of people. An estimate in Japan, 20 million people have seen these films that we've made about the dog macaques. It's actually sad statistics. It's enlightening. It's satisfying to me that so many people in the world, millions and millions of people, have learned about the wonders of macaque society, about primate behavior, about the evolution of it. Sadly, in Sri Lanka, some people don't even know that we have 2 different or 3 different species of primates. A person watching TV in Chicago will know more about primate behavior than a resident of Polonnaruwa, which is sad. And what's the solution to this? Well, we have out of the 40 documentary films that we made over the last 30 or 40 years, 2 of them are translated, are narrated in Sinhala. One of them is very popular. It's called Tempu, narrated in English and also narrated in the Sinhala language. And the Rupa Vahini has shown it almost ad nauseum, or sections of it over and over again. So it's popular. So it brings an awareness to people that monkeys are something more than just a pest in your garden. There's something we can do about it. So raising the awareness is one thing. The other thing, of course, that we have to deal with is this human-monkey conflict. This is probably the main paper that we put out in 2019. It won the Editor's Award. It brings the problem of human-monkey conflict into perspective and what we can do about it. Of course, I don't do this work alone. This is my team at Polonnaruwa. My dedicated team at Polonnaruwa, and these are the institutions that I'm indebted to for support. And thank you very much.
Thank you very much, Presidents. It's an excellent presentation. It's a question time.
So I have a brief comment and a question. When I came to know that he was from Smithsonian, I asked him whether he knows Sue Ripley, who I had met when I was 3 years ago, 3 years of age. So it's a small world. My question to you, wearing my ethics hat, international ethics hat, we take ethical clearance when we do human subject research. So monkeys are very close to human being. It's a difficult question, I know. What are your thoughts about ethics and research among monkeys?
Well, I don't do laboratory studies because I think it's unethical. And you can learn more about monkey behavior, animal behavior by just taking a notebook. You don't need sophisticated laboratory equipment. What you need is a brain, most of all, a piece of paper and a pencil. And a pencil is better than a pen because if it rains, your pen will run. So yes, and just develop, observe, develop your questions, develop a protocol to answer those questions, and slowly your knowledge will build up. That's, yes. In fact, I really don't like people putting collars on elephants. You don't need to put a collar on an elephant. Elephants are so individually different, distinct. They have different tails. There's so many individual markings on an elephant. Anybody who wants to put a collar on an elephant or on a species, I think they should be flunked. They should not be given that research. Thank you.
Okay, thank you. Like we are trying to, in fact, one project working on a color, but again, like we want to, like a main looking at the, like a main culprit, like we can't kill them. And, we want to have a kind of a control. Otherwise, yeah.
But you can follow them on foot. By zoologists, you follow them in Galai on foot. Sometimes, of course, you have to do the jeep because these things walk 30 kilometers a night. But anyway.
Okay, thank you.
Okay, over to the next speaker, Professor Adhikaram, Natural Plants.
Actually, I'm going to talk about some post-harvest problems. Although I work on the natural products section. Post-harvest disorders and diseases of mangoes. That's my topic. In the cultivar from EJC, which is currently exported from Sri Lanka. In fact, the variety has established well in the export market and they're expanding. Now they have certain disorders and diseases, and we are studying them under research project, NRC project under the category public-private partnership, and they export. He is our industrial partner who bears 50% of the cost of research. And most of the work that I'm going to present here was conducted under this project. And the work was conducted here at the NIFS and the University of Peradeniya, as well as 11 horticulture plantation. And we have two co-investigators, Professor Lanitya Singh and Professor Devan, who does our molecular studies at Peradeniya University. And I must say that we have signed a non-disclosable legal agreement with the donor. Therefore, some of the findings on the project cannot be presented in this presentation. Now, just to refresh your knowledge, diseases and disorders. Now, diseases are generally or normally caused by biotic agents like fungi, bacteria, mycoplasma, etc. Disorders are caused by abiotic factors and not by biotic factors like nutritional, environmental, cultural factors, and sometimes post-harvest handling or pre-harvest handling can lead to disorders. And disorders are not transmissible. Diseases are transmissible, but not the disorders. And the purpose of the work is that now disorders and diseases lead to post-harvest or fruit losses here, or sometimes they lower the market quality. And here the diseases and disorders that we are interested can collectively cause losses up to 20 to 25% of the harvest, which is quite a lot. And the purpose of the study was to diagnose these disorders and diseases, understand their causes, study the mechanism of development, and also the factors affecting their development, and finally develop appropriate management practices using all information gathered. In this particular project, the ultimate aim is to increase the availability of good quality exportable fruits. There are many disorders that affect mangoes, but I'll be talking about two disorders which are more common actually. Usually disorders are in fruits are rare, but these are common at least in this particular variety. Now this one we call it internal pulp browning, which is a new disorder. The cause is unknown, not recorded anywhere else. And here this was first observed in 2012 in the same variety. And the grower brought materials to me in 2012. I did not really do much work at that time. The main symptom is when you cut the fruits, you will see browning, like in the photograph, in the pulp just outside the seed on both sides. On both sides, but where from outside, before cutting or from outside, there's no sign of the disorder until you expose the food pulp. So this kind of disorders can create an element of uncertainty in the mind of suppliers as well as the consumer regarding they don't know actually the actual food quality. Sorry, sir. And actually, observations we made, in this particular variety, the seed begins to germinate while inside the fruit, which is not normal, and it produces sometimes the spinules and also roots also sometimes. That's one observation, and also a bacterium could be isolated from this affected part. Right? But later on, we found that it has no direct role on the. Okay. And now, thirdly, this disorder occurs only in the fruit season from October to December. Right? So that season, during that fruit season, they get a lot of rainfall. But the other main reason is season is from June to August. Now they are harvesting, but they never get this disorder during that season. So we have built up a hypothesis. Now, this seed germinates inside while inside the fruit, and then when it germinates, it swells. It increases in volume, and then so we think it normally you can see that in not the wheat, but the height is it? I don't know. It expands that really exerts pressure on the pulp on either side, so that may lead to tissue destruction, and then phenolic compounds and enzymes are released, and oxidation takes place and browning happens. And we have shown that the seed thickness increases during germination. We have taken out seeds from 50 seeds with germinated, 50 fruits with germinated seeds, right? And then we have compared them with ungerminated fruits, and we found that during germination, seed swells twice. It doubles the thickness. I have some more. So this rainfall actually we have monitored from 2012, and we found that it's also related to heavy rainfall. We don't know the reason, but we think that it helps seed germination, promotes seed germination. And also we have analyzed pulp, and we didn't see any differences in the levels of macro elements or micro elements. So that work was done at the IFS. Professor Rohan Virusuriya provided this ICP facility, and also academic spectroscopy. It's another disorder which we studied here. We call it picking. You see these patches. Those are concave kind of areas. Sometimes you get one or two, but in some fruits we can get large numbers of these peelings. And this is also not recorded in Sri Lanka, but it has been recorded some other fruits. And peeling appears now when you harvest the fruits, you can't see any peeling. But hours after harvesting, these appear. Now we did some anatomy. We examined a section through these peelings, and we find actually some tissue layers, about 10, 11 tissue layers below epidermis. They collapse, right, and compress. That's like this concave fillings here actually. And this disorder is related to deficiency of some elements. And we again actually have done some micro elements. We analyzed first at the ITI and then at the NIFS again, Hans lab. And then we found actually 3 elements, 1 micro element and 2 macro elements. They are actually excessive. We always found those elements more in the filling tissues than the controls. And then also we have tested EDX in the scanning electron microscope at Peradini, and we found the same results. And then we had another plantation at Isamaharane, and they had this filling in the same variety in plenty. And actually, what we found later on, actually, we found a fertilizer mixture with the 3 elements that I have mentioned. And the moment actually we recommended them to use those that fertilizer mixture, and then fitting percentage was 12 in 2012, and then actually anyway, so then actually that really managed the problem. So I will go into just one more thing. Actually, we were also doing some microalgae work. In microalgae work actually, in addition to this post-harvest work, actually we have prepared a checklist of plant pathogenic fungi and Umai in Sri Lanka, consisting of 400 species which were recorded from 1950 to 2000, and also we have provided some information to the National Red List of Sri Lanka through Professor Vijay Sundara, and the fungi associated with plants in Sri Lanka, again the fungal list, and also a flora in Sri Lanka, and also we have found some new fungal records. Thank you very much. Sorry for taking
Thank you very much, Professor Adhikaram. Unfortunately, we don't have time, but we need to quickly move into the next presentation. The next, I would like to invite Professor Benjamin to talk about his evolution, ecology, and biodiversity project. Thank you.
Hi, good afternoon, everybody. I think the special forces in science are remaining, everyone else is gone. So let's see. I'll try to keep you awake for the next 8 minutes. So I have subtitled my today's presentation based on the new normal, vistas of biodiversity and spider splendor. Okay, here we go. So Professor D just talked about the importance of biodiversity. So the world's wealth of species is called biological diversity, and it represents the basis of mankind's existence in our planet. Biological diversity comprises not only animals, plants, fungi, and microorganisms, but also their genetic variability and the ecosystems they live in. So why spiders? Spiders are diverse, abundant, important, and beautiful and relevant. So here we see that when you take insects and spiders together, they form a large part of the biodiversity. If you take mammals, it's like all mammals would be about 0.1% of the biodiversity. Sorry, Professor Dieters. So, I mean, all what has been done by my project is in the annual review book. So I just give you a small abstract. One postgraduate degree was awarded during last year. 6 research papers, 4 in index journals and 2 in other. 12 new species were described. And my research was highlighted in international media as well as the local media. One paper was in the news first prime time during COVID-19, when everyone is home watching news. So that was really a good publicity. So basically, I mean, I use molecular phylogenetics to study spiders. So what you see here is we can group related species together. So you see like me and my sister is close related, and you see that in one branch, and then they're more close related to each other, and then you see the cousins in the next branch. So if you have something missing, and you combine it to this phylogenetic tree, you can probably find its spacing. So some of you have wondered how we know that the COVID virus that is in humans, where it came from. You know, someone I wondered. That is done by molecular phylogenetics. Here is I got this from the internet. Here you sequence all the COVID-19 virus from various animals like pangolins, bats, etc., and run the relevant analysis to find its closest relative. So we use similar techniques here on endemic spiders. So these submitted thesis, I guess the review has come. I hope that we can have the defense this month or next month, next month I mean. So it's by Delaney. Title was molecular phylogenetic placement of jumping spider genera. This is one part of there are 3 chapters. There is one section of that. I can't see it clearly. It's about we found about 6 new species, mostly from the Central Highlands. Then I will move on to what we are doing currently, also started last year. Unfortunately, we didn't receive much capital or recurrent funding, so most of our work has been partly stopped. The molecular lab is not functioning because we didn't receive recurrent funds for last year and this year. So I'll just show this is the work by Abhira, and she's working on also an endemic genus of jumping spiders. Here we actually have 3 described species. We have found another 2 new species, but I was supposed to go to Paris to a museum to figure that out, but which are the new, we are not sure which are the new species and which are the ones described. So I was supposed to go to the Paris museum where the types are, but unfortunately, due to the COVID-19 lockdown, that has been postponed. There is work on stick spiders, a very interesting group. There are 2 groups related. One live in webs of other spiders, and others live freely, hunting on their own. The spiders that live in webs of other spiders, they steal prey from the owner of the web. So it's really interesting. We are interested in the behavior and how it evolved. So it turns out that there are at least about 4 new species here, the 6 spiders, and all those are like endemic and new to science. Here you see images of those four new species, and these are preliminary phylogeny we have actually a phylogeny where we included all the Sri Lankan species in a worldwide phylogeny of this group of spiders. So we are still working on this. And thank you very much. And this is my team and logos of some of the funding agencies. Thank you for your time.
Thank you, Professor Benjamin. Like we have maybe three questions, 3 minutes. Shala has one, maybe it's not very much directly related. Like you have done a lot of polygenic work, and you have done like identified over 100 species. Like where we can go, where can push into the next, like what you want to do, like is a so goes on identifying new species, so like so you want to go into something, push into something deeper, or like want to go into something other application side?
No, actually, we have discussed this question before, I think, because in several forums with you I have discussed. And the major issue is, funding. I mean, if we can, and I have some international funding. Last year I received like 4 million from the German government. And so what is the funding is the government giving us? I mean, if they are willing to give funding, if it's important to the government, yes, we can move forward. But if the I don't know, according to the IFS, I mean, we haven't received any capital money. We haven't received only salaries for recurrent. So all my ideas are stuck up somewhere. That's the answer.
Thanks, Professor Benjamin, for bringing that issue. I mean, it's an issue we have identified. I'm not 100% sure whether this is the right forum to discuss about it. But I can assure you one thing. We look into that. I totally agree with you. Without funding, we can't do anything. And funding strategy is as equally important as research strategy. So this two should go in hand. I can give an assurance. We will seriously look into that and explore the way. And I can assure you, your research will go ahead.
Thank you very much. That's very encouraging, Mr. Tom.
Now you scored a century last week, right?
It's actually a personal goal. I described 100 new species. So
on an average, per year, roughly how many new species?
Well, that depends on a lot of things. Normally about like 10 to 15.
So these are like 100 is over a period of 10 years or so.
Yeah. So I mean, it's like to be put it like in a worldwide context. Then I have my colleague who is in Bonn, who is actually my collaborator. Yeah, he has reached close to 1,000. So I mean, so it's like just a relative number. So,
thank you.
Thank you, Professor Benjamin. One more question.
Just simple question. Well, Benjamin is quite cancy at night.
So there are different families. There are like wolf spiders that see in light. They have excellent vision in night, and there are others. I mean, web builders, they have poor vision, but they use sense. I mean, like the web is like a extended body where they're like a extended sensory organ in the web. So they have less vision, but they have more organs to sense the vibration of the web, and like go to their prey, and then mating, and they can recognize mates that come onto the web and things like that. But they have less vision, more other kinds of sensors.
Thank you very much, Professor Benjamin.
Last but not the least, important thing, Dr. Bowatkar is going to talk to us about environmental science research program, air pollution and risk assessment.
Good afternoon, everyone. So I'll try my best to keep within the 8 minutes. First, before I start the presentation, I would like to thank the Research Council and Director of the NIFS for giving me a visiting research fellowship, which I finished my after leaving the NIFS in 2019. So what I'm going to present here today is a continuation of the research that I have started in 2018 March. So it gave me an opportunity to continue my work here. So this is an immature project, so started just 2 years ago, so you wouldn't see much depth results that's what we have seen in the previous research presentations. Yeah, so my main work is related to air pollution and diseases. Most we know that air pollution is a global issue. So most of we are exposed to air pollution, and there are a lot of diseases emerging from exposure to air pollution, and especially in Sri Lanka, is a neglected health issue. So we don't care much about air pollution. But if you go to the other countries, like developed countries, air pollution, their levels of air pollution is much lower, but they are still taking higher. They're doing more research and more awareness and try to mitigate and reduce air pollution because they know that air pollution can cause a lot of health issues. So, so according to the WHO, World Health Organization, the air pollution levels in Sri Lanka is relatively high. But the issue is in Sri Lanka is we don't have a regular monitoring mechanism that should be ideally done by the Central Environmental Authority to fulfill some of the gaps of the monitoring. So we thought of establishing a monitoring network to measure air pollution in Sri Lanka, especially in the major cities, and to get an idea about the levels of air pollution and what type of link with the air pollution has with different diseases. Yeah, so this research project is aimed at monitoring air pollution using advanced smart sensor technology, real-time air pollution monitoring and data visualization to model the air pollution, estimate health risk associated with air pollution, and also evaluate performance of air pollution control methods. So these are the long-term goals of this research project. So we use smart sensors. This project is a collaborative project with the couple of universities in Australia, mainly the Queensland University of Technology. The reason for using the small sensors is that the standard monitoring stations are very expensive. Like the standard monitoring station could be like over 20 million rupees. So that's very expensive. So the new technology, these smart sensors are coming up. They are relatively cheap and they can do the job to a certain extent, like what the standard monitor is doing. These are small sensor units. It can measure particulate matter and carbon monoxide. And also these are solar powered sensor units, and it can communicate using internet. So that's one of the really advantage. We can see the actual air pollution levels real time. So what we measure is the small particular size. These are, if you can see this picture, so these particles emitting from different sources like from vehicles and burnings. So these tiny particles here, so this is a picture of a hair, human hair. And so these tiny particles go up to, this is a PM 2.5, I guess, here. So these tiny particles can go into humans' lung, and it sometimes can go to the bloodstream and cause a lot of health issues. So these are the main particle pollutants that we measure. So this is an image of the sensors that we have in different places of Kandy and in Colombo. And so we can see the real-time air pollution using this online tool. So this is not public yet. So we are trying to make it public, and then people can actually see what the air pollution levels they are exposing to. So, so we have done some measurements over the last year. So this is this is the standard monitoring station recently established in Kandy in the Torrington Square. So it's a big one, but our sensors are very small. So these are the ones that we kept to see how best our sensors are working against the standard monitoring station. So this one shows the 1 year worth of data. I think Kandy, for best of my knowledge, this is the first time we are talking in a scientific forum about long-term particulate matter concentrations in Kandy. So you can see, so this is the month starting from January 2019 up to December 2019. So this is mainly my research assistant, Mr. Mahesh Senatapna's work. He has done all the hard work to get this work up to here. So we can see that in especially the air pollution, the PM 2.5 rising up to March, then it's going down. I think this is mainly due to the change in the weather patterns, because we get a lot of rain, so the particulate matter can be washed out by the rain. So this shows the average daily particulate matter variation in the Kandy. So if you get a typical day, so this is the concentration what you see. So this is the time in this x-axis, and y-axis the particulate matter concentration. So in the morning we see a big peak, then it's going down, and then we see another small peak in the afternoon. So we got the opportunity from the data that we got from our sensors to look at, if you can remember, there was a huge air pollution event occurred in 2019 November, if you can remember, in Kandy and most of the country, even in Colombo, the cloud of air pollution was there. So this was hypothesized due to the higher pollution levels occurred in India. So this was some of the images taken from satellite and developed the air pollution levels. And you can see that this is the 1st of November, this is 3rd of November, and this is the 6th of November. You can see that the air pollution is coming from North India to Sri Lanka by wind. And it hit Sri Lanka around 3rd of November, and we observed a very increasing air pollution in Kandy. So these are the results from 8 sensors that we had in Kandy. And this is a picture from my home, so I'm not sure whether it's clear. So this is in a clear day what you see in the sky, and this is giving the air pollution. And I thought of presenting some data due to the reduction of air pollution related to COVID-19. So this is actually not related to 2019, but for the interest, and also if I'm going to present this in the next year, the value of this may be diluted at that time. So just for that, for your information, so we took measurements during the curfew and before the curfew in Colombo and Kandy. So we observed that there about average 30% reduction of PM 2.5 in Kandy, and about average 41% reduction in carbon monoxide. And also in Colombo, it's about 50% for PM 2.5 and 54% in carbon monoxide. So this is graphically the air pollution change in Colombo and Kandy. So before is the red color one, and during is the green color one. These are the two projects. We wanted to look at the health effects as well. So we looked at the household air pollution exposure and allergic and respiratory disease in children. So I'm not going to talk in detail, but briefly we saw that houses which had the biomass fuel, they had an increased risk of these, and also eczema. Eczema is one of allergic diseases. And we also looked at how the air pollution, household air pollution can affect the adverse fetal growth. We saw that how the biomass fuel can has a major effect on low birth weight. So I'm not going to talk in detail. So these are the publications up to 2020 up to now. So 15 peer-reviewed publications, about 14 abstracts, some my international collaboration work as well. So this is our team. Mahesh, Sajid, Sampath, Diluksha, main core team, and this is an Australian students who came last year to conduct the household air pollution work. They completed their work and left the country a couple of months ago. Thank you.
Okay, thank you, Dr. Gayan Govardhan. We have time for one question.
People grow that air pollution. Well, how do you make that connection?
I'm sorry, I didn't get the question.
Growth infects and air pollution. How do you make that connection?
Can you repeat it? I missed the first part.
Okay. You indicated that fetal growth or infant growth is impacted by air pollution. Is that because less oxygen for the mother, or what's what are the intermediary steps?
So you know that one could be the reason of the building up of the carbon monoxide. Carbon monoxide can bind to red blood cells, and the pass of oxygen can be reduced to the growing fetus. That can be one hypothesis. And the other one is that so there are a lot of toxic compounds can be absorbed through the lungs to the growing fetus, and those toxic compounds could affect the growing fetus as well. So there could be not a single mechanism, but there could be multiple mechanisms involved.
This is across all sorts of strata.
Yeah. Did I answer your question?
Yes, you did.
Okay. Yeah, thanks.
Thank you very much. Let's show our appreciation.
So thank you very much, Professor Atul Sumitpala and Professor Saman Selimvira for sharing this session. Okay. So I invite the chairpersons to give conclusive remarks.
Thank you very much. It's customary to give a summary of today's presentation, which I'm not going to do because we're already late. But let me say very few words about it. I really enjoyed the whole thing. It was a lot of learning for me as well. And I think looking at it, listening to it, extensive amount of work has gone in, which is very difficult to present in 10 minutes. It doesn't do justice. But unfortunately, that's the challenge and the discipline. However, we are 45 minutes late. So I take full responsibility and apologize to you because I have failed in my duty as the chairman in this chairing these 3 sessions. And that will not happen next time, I assure you. So we'll start on time and finish on time. But it was great. I have to say that, and thank you very much, really enjoyed. Thanks to all the presenters who has done your work and presented here. And I look forward very much to participating in this type of things. And I actually want to come and spend more time with you all, learning more about what you did. And particularly when Professor Jesundar presented about Arboretum. We visited there, we listened to it. More I listen, I know more to learn. So everybody. And I should also make a special thanks to our director, Professor Senavira, for doing this. This was excellent. And I'm sure there is a lot of efforts behind this. This type of a one-day conference is not easily done. So thanks to everybody who has done it from the front as well as from the back. And really enjoyed. Thank you very much. I'm pretty sure our director has few words to say. Have a good day. Good night.
Thank you very much, Chairman. Again, I want to say, I didn't expect to have the magnitude, and it's excellent, and it's more than expected. And you all did a lot of hard work. I really want to congratulate for the team, especially Professor Gamini, Senator who lead the team, and also all the members, Professor Bandar, Dr. Ratnayak, Dr. Jayaratna, and then Professor Malikar, Dr. Lienage, and Dr. Tilakaratna, and also our SCUD team is Gayan especially, and also photographist team again Gayan, and the main Senaviratna, and the whole SCUD team, Viraj, and if I miss anybody, I'm really sorry, but again, it was an excellent job, and especially Suvain, she took the responsibility to manage the whole the conference, and Dr. Shalini, and I miss out as I know Shalini did a whole lot of work. And did I miss out anybody? Like if I miss out, I'm very sorry, but again, so you done a fantastic job. And again, the whole institute supported you, and so all is a congratulation. It's well done, well supported. And I pick up 2 things during the conference, so I always, I thought, next meeting, next conference, so we need to pick up, this time we got a new award system, 11 people award for the service award, and also the professorial, like the publications award, and also we have a 3-4 category of the achievement award. I think we should pick up next year overall achievement award. We need to, especially when I listen to the Professor Lakshman and how much, not only the science and the public and the institution building, that type of thing is we should recognize. That is, we don't have here, but I think next year will be another award for the person who do for the institution building and national building. This is very important. And other thing I pick up is that we have the research, the area of like solar research. Actually, this institution is very famous, and we were in the top 10 in the world, like 10, 15 years back. But we have several groups working together, working in the same thing. It's a high time to set up a National Center for Solar Research here at IFS. That should happen, I think, soon. So we will think about that one. We need to team up, like we discussed, and get into research council and board, and then we should establish that one here. That will be the best research center of this institution. And it is, I really want to say, great, thank you, enjoy, and it's a wonderful meeting. And also, we should think about leadership building annual as well. That's we need to discuss a little bit later. And we do a lot of work, but we also need to do the leadership in the sense that academic leadership. We are all academic, belongs to that family. And we should have that, we should build. And otherwise, it's fantastic. I don't have any words to say. I really want to celebrate after, but again, we don't have much time. We're already late. And I will stop from here. Again, thank you very much. Really appreciate it.
Thank you very much once again, Professor Atul Sumitipala, Chairman of NIFS, and Professor Samal Senuvira, Director of NIFS, for your thoughts and also chairing the session, final session. So as we end the program for the day, I would like to thank all the presenters as well as all of you for being present today and for being a wonderful audience. So while appreciating the great effort by the organizing committee of the event, we end the annual research review of the National Institute of Fundamental Studies 2019. I wish you all a pleasant evening.